So we've brought up acid rain before in this class in the context of sometimes acid rain can get into soil and trees and lakes, and that can prove toxic and hazardous for the life in that soil and in the lakes. Okay, so today we're going to be talking about titrations and how that is so important for something like acid rain, which can infiltrate these different environments. So we have this figure here to start off. Acid rain can begin generally, so in industrious places that are creating a lot of kind of emissions, such as sulfate and nitrate. So this pathway kind of showing as it travels into the atmosphere, the pollutants are transformed into acid particles. Okay, and that's going to be transported long distances. It's going to be absorbed by the water molecules in the cloud formations. And then when it rains, those toxic and acidic molecules like nitrate and sulfate are going to fall to the earth, and they're going to be collected by the soil and by the lakes. And if acid rain collects or is collected in enough lake, the fish can die. So this can also be true for the plants and the trees that grow if the soil becomes too acidic. So scientists often can take samples of lakes and they can test the acidity of them. And they can test the acidity of lakes through this idea called a titration. So for this, acid rain creates unwanted chemical changes in soil, causing damage to trees and other plants. And so scientists take water samples and they want to test the hydrogen ion concentration, right? Because an acid is basically one that has accumulated a lot of protons, a lot of hydrogen ions. So for our basic definition of titration, titration is a neutralization. Let's change the spelling. Neutralization reaction. And it's monitored with an acid-base indicator. Okay, so generally we'll have a acid with an unknown concentration, and we want to figure out how concentrated or how many hydrogen ions are in that solution. So if we look here on the right, we have a titration setup. Okay, so the long thin tube on the right, this is called a burette. And basically you have this handle and you kind of twist it like you would how you would wind up a toy. And as you twist it, the base, which is found in the burette, is going to slowly start leaking out into the solution with the acid. So during night titration, a base is added from the burette until the indicator changes colors. So we're going to watch a brief example and from this video, and we'll kind of go through it together. All right, so I kind of jumped ahead. Okay, so you have this base, you have an acid in this beaker, and then you combine it with water, you rinse it off, and then let's see if we can get to the titrating part. Okay, so you kind of turn the handle on and you basically start mixing up the solution as the base is being added. And then there you can start to see the color is changing, and then it starts to slowly dissolve back into the solution. So you're looking for a saturation point where the base has fully dissolved in the solution, and then you start to have a color change form. Okay, like that. And then there you can kind of calculate, okay, I added this much base, maybe I added 50 milliliters of a base, and then you can figure out, okay, this is how strong of an acid I have. So you can use it to calculate how much base you put in, and from there, find how much acid you have. Okay, so if we look at a particle view of titrations, we have this equation, hydrochloric acid plus sodium hydroxide turns into sodium chloride and H2O. So if we say we take 2 drops, one that has hydrochloric acid, and those are our acid particles, our protons, and we have a base drop of sodium hydroxide, where the blue is our hydroxide ions. We have the same amount, 1 molar molarity for each one. We add them together, and basically, so this one, figure 3, as equal volumes of 1 molar of hydrochloric acid and 1 molar of sodium hydroxide are mixed together. And so these ions have actually decreased, like, right, are free-floating ions, because each ion has spread out over twice the volume, and that's equal to, let's see, half or 1 drop of solution, or sorry, 2 drops of solution. Okay, so the next one, that one was equal amounts of acid and equal amounts of base. This one, if we look, we have the same amount of acid as the previous one, but now we have twice the amount of sodium hydroxide. So in this figure, figure 4, we add this together and we can kind of see what happens here. The base is more concentrated than the acid, so the 2-drop mixture will have leftover sodium hydroxide, right? That's what we can kind of see on the right-hand side here. Because the 2.0 molar sodium hydroxide is doubled, or twice as concentrated as hydrochloric acid, you only need half as much, right? Because we have a lot of leftover hydroxide ions floating around. And this table kind of shows that same idea. When we add things in the same amount, it should take about the same amount of drops to cancel each other out. And when you have twice as much, it should take half that amount. Okay. Basically, equals, that should take the same amount. And if you have twice the amount, it should only take half the amount of volume. All right, so the next video will go over the calculation part. I'm gonna split these videos up, and I'll talk to you guys soon.